Hi, my name is Campbell DeLuca, and my research serves to quantify the effects of glyphosate on the neurological, behavioral, and reproductive behaviors of aquatic organisms. The herbicide glyphosate is the main component in the popular product Roundup, which is a weed killer. In recent years, the use of Roundup has increased both agriculturally and commercially, which has had devastating effects on the world around us. In fact, recent studies have considered glyphosate to be a high-risk herbicide for underwater organisms. These devastating consequences are a cause of the lack of knowledge that surrounds glyphosate and how this lack of knowledge causes humans to abuse glyphosate. By spreading awareness about this topic, we will be able to help humans foster a better future for both their world and the underwater world. In previous studies, researchers have only tested the basic effects of glyphosate on aquatic organisms, such as survival rate. In these past studies, it was found that as there was a higher concentration of glyphosate in a system, the survival rate of the organism decreased. My research serves to go farther by testing how glyphosate is able to further impact aquatic organisms. To do this, I will quantify the change in expression of serotonin and the change in expression of the female vitellogenin gene in male crayfish when crayfish are exposed to higher levels of glyphosate. In my experiments, I'm using 3 different strains of crayfish, which is 12 in total. And the crayfish are lobster-like organisms, which are going to be used as the model for aquatic organisms. So there were 4 crayfish in each tank. The first tank was the control tank, which contained 4 liters of salt water. The second tank was a control or experimental tank, which contained 0.33 micrograms of glyphosate with 4 liters of salt water. The third tank contained 0.66 micrograms of glyphosate with 4 liters of salt water. The amount of glyphosate used during the exposure was calculated by understanding and studying the median amount of glyphosate, which was measured in Midwest streams and rivers in past years. And this will allow us to kind of keep a real world view on this and how how aquatic organisms are affected in the real world. Throughout the month-long exposure period, crayfish were monitored 24/7 utilizing a video camera in order to track behavior changes. And throughout these observations, it was found that in the experimental trials, crayfish had significantly higher energy and were therefore more hostile towards each other. But in the control trials, the crayfish did not interact with each other and had very low energy. As of right now, I have performed qPCR, which will allow the quantification of the changes in expression of serotonin and the changes in expression of the female vitellogenin gene in male crayfish. And as a continuation of this research, I plan on utilizing the delta-delta CT analysis method in order to calculate and quantify the results of qPCR. And this is by calculating the relative fold gene expression of the samples. The data I have collected and will collect will serve as major findings in my research and understanding these effects of glyphosate on aquatic life.